All right, guys, we are back again, and we are going to talk now about sex-linked traits, pedigrees, not the dog food, and dihybrid crosses. So we've got a lot to cover today. So we are still looking at Georgia Standard of Excellence SB3B. So we're analyzing how biological traits are passed on to successive generations, and we need to be able to use mathematical models, those Punnett squares, to predict and explain patterns of inheritance. Another thing that we're going to learn how to use are pedigrees, and those aren't necessarily going to predict, but they can help explain. So you should be able to analyze a pedigree and also analyze a dihybrid cross by the time that we are done with class today. So, some inherited traits show up in males far more often than they do in females. So, we know, we've talked about this in class, that humans have 23 pairs of chromosomes. 22 of these pairs are what we call autosomes. That means that they're part of your body cells. They make up things like your hair, they make up your guts, they make up your heart, they make up your muscles, they make up your lungs, they make up your kidneys. So 22 of these pairs have to do with that. One pair is what we call sex chromosomes. So sex chromosomes are the chromosomes that give you your biological gender. Everybody has one of two biological genders, that is XX and XY. Some traits are carried on the X chromosome. They're called X-linked traits. There are very few traits carried on the Y chromosome. Guys, size-wise, the X chromosome is about, oh, hold on, let me turn on my pen. There we go. Size-wise, the X chromosome is about this big and the Y chromosome is about that big. My little red marks there. Y chromosome is really, really small. It really and truthfully, the most important gene that's found on the Y chromosome is the SRY gene. And the SRY gene is what turns on testosterone so that a person will develop male sex characteristics, both secondary and primary sex characteristics. Everything else that's carried on that pair of chromosomes is carried on the X chromosome.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cells that help stop us from bleeding. They help our blood to clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets, and therefore the ability of blood to clot. With hemophilia, even a small cut could be dangerous because the bleeding can be continuous. There are now many good treatments for the symptoms of hemophilia that have greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it is different from general Mendelian genetic problems. With sex-linked traits, we still use the terms dominant and recessive for alleles, except this time, those alleles are on sex chromosomes. This is the case with sex-linked traits. What is a sex chromosome? Well, first take a look at this karyotype, which shows a human's chromosomes. This karyotype has 46 chromosomes arranged here in 23 pairs. 46 is the general number of chromosomes in humans, but as we mentioned in our karyotype video, individuals can have more or fewer than 46. Chromosomes are made up of DNA and protein. They contain your genes. 23 of this person's chromosomes came from an egg cell, and 23 of this person's chromosomes came from a sperm cell. The last two of the 46 chromosomes in this karyotype are called sex chromosomes. The sex chromosomes are commonly called X or Y chromosomes, but X and Y have nothing to do with the shape of the chromosome. The reason for the X and Y names is really interesting. Check out our further reading to learn more. All humans have at least one X chromosome. In most cases, females have a combination of XX and males have a combination of XY, but individuals can have additional or fewer sex chromosomes. We're focusing on sex chromosomes in this video because we're going to show how to do Punnett squares with traits on these chromosomes. In the case of Punnett squares with sex-linked traits, the traits being tracked tend to be on the X chromosome and not the Y chromosome. The X chromosome is much larger than the Y chromosome and contains more genes than the Y chromosome, so technically this video will be about X-linked traits, but we do want you to know there are Y-linked traits too. Referring back to the beginning, when we mentioned the disorder hemophilia, hemophilia is a sex-linked recessive disorder carried on the X chromosome. So focusing on that, let's learn how to solve a Punnett square that involves this trait. We will use the capital letter H to represent an allele for not having hemophilia, and a lowercase letter h to represent an allele for having hemophilia. Why? We're going to do that because we mentioned hemophilia is a recessive sex-linked disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the X chromosome as a superscript, like an exponent. Let me explain what I mean by that. Let's consider a female individual shown with XX here. If the genotype is X big H, or X big H, X little h, this female will not have hemophilia. Why? Because as long as there is at least one dominant allele, that dominating allele will be what shows in this trait. So no hemophilia. Hemophilia is a sex-linked recessive disorder. However, the X big H, X little h genotype would be a carrier, meaning while the person wouldn't have hemophilia, they would be carrying the recessive allele. The only way for this particular female to have hemophilia would be the genotype X little h, because only when there is no dominant present will that recessive show up, at least in this type of trait. Let's consider a male individual, shown with XY here. X big HY would be a genotype of a male without hemophilia. Notice how I didn't put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If this male had the genotype X little hy, then this individual would have hemophilia. Since there aren't two X chromosomes for this person, either this person has hemophilia or doesn't. Let's try a Punnett square problem with this trait. Let's say these two people here both do not have hemophilia, although the female is a carrier of hemophilia. If they together have a biological child, what is the percent chance of the child having hemophilia? Also give genotype and phenotype ratios. Step 1. Always determine the genotypes of the parents first. The female must be X big H, X little h. Why? The Punnett square problem says the individual doesn't have hemophilia, but is a carrier. That means this heterozygous genotype. It says the male does not have hemophilia, and so the genotype must be X big HY. If it was lowercase h, this male would have hemophilia. Step 2, place one parent's genotype on the top, outside the square like this. Place the other parent's genotype on the left, outside of the square like this. Step 3, fill in the square. For formatting purposes, place X chromosomes before Y. You also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. And the phenotype ratio, remember that these are the traits, can be written out like this. There's a 75% chance that a child will be born without hemophilia and a 25% chance that a child would be born with hemophilia. Remember, like all Punnett squares, these are representing probabilities. So because it's a probability and not necessarily the exact outcome, they could, for example, have quadruplets that all do not have hemophilia, or they could have quadruplets that all have hemophilia. The probability of the latter is less likely, but
All right, guys. So, um, we're going to look at some examples of our sex-linked traits. So, we know that everybody inherits at least one X from mom. Sons get an X from mom and they get a Y from dad, just like they said in the video. Daughters get an X from mom and an X from dad. So, dad is actually the one who determines biological gender. In order for a daughter to have a sex-linked trait, she has to get the recessive trait from both of the parents because she does have a second X. Whoops. I don't know why that did that. Weird. But she does have a second X that will allow her to be able to mask the recessive trait. Men, on the other hand, only have one X, which means they only have one allele that carries the trait. So, because of that, males don't have a way to mask it. If they get the disorder, if that trait is on their genes, then they have the disorder. And there's no way to get around that. Now, an example of X-linked traits are color blindness. One big example. So, 1 in 100 females will be colorblind, where 1 in 10 males will be colorblind. Because remember, males only have one X chromosome and one Y chromosome. So, there's no blocking it for males. Something's not right with my Apple Pencil. Females, on the other hand, have two X chromosomes. Because they have two X chromosomes, they are able to block or hide the disorder if they happen to have one affected chromosomes. X-linked traits are only expressed in females if there are two copies of that X-linked traits. In males, there only has to be one copy of the recessive allele for it to be expressed because they have no second allele to mask it. If you can see the numbers here, you are not colorblind. If you cannot see the numbers here, you might want to check with your eye doctor. So now we're going to talk about pedigrees.
In this video, I'll walk you through how to look at a pedigree and understand what it's showing you. A pedigree is like a family tree in that it shows you relationships between family members. We can use a pedigree to trace a trait through generations. Generally, the trait in question is a genetic disorder like hemophilia or sickle cell anemia, but you can use pedigrees to trace any trait and determine how it's inherited. So what are we looking at here? In these charts, males are represented with squares and females are represented with circles. A person of unknown sex is represented as a diamond. Here we can see two biological parents connected by a mating line. From their mating line, you see a line of descent that leads to a sibship line. The sibship line connects all of these parents' offspring. The children of these parents are listed in order of their birth from left to right. So these parents had 3 children, a son followed by 2 daughters. Each generation is noted by a Roman numeral. You can see generation 1 here and generation 2 here. And each generation has individuals labeled with an Arabic number. So we have generation 2, individual 2, who in this pedigree is female. If you want to note that a person in the pedigree is deceased, you'll just draw this kind of diagonal line through them to indicate that. Now, tracing traits through a pedigree requires you to know which individuals have the trait and which individuals do not. A shaded circle or square is a person who has the trait. We'll call this individual affected. And an unshaded circle or square indicates an unaffected individual. So if we look at this pedigree, we can see that the trait we're tracing appears in the mother and in her son. But the father and the 2 daughters are unaffected, meaning they don't have the trait of interest. Now, when a person mates with someone outside the family, you connect the pair by a mating line, like this. So this female mates with a male outside the family and has 2 sons. These sons are in generation 3. Let's take a look at a larger pedigree and talk through what we're seeing. Here we see generation 1. This male and female produce first a son, as you can see here, and then a daughter, as you can see here. This son mates with an unrelated female, producing 3 children, 2 daughters and 1 son. Now what we see here is that this female from generation 2 mates with an unrelated male, and they also have 3 children, a daughter, then a son, then another daughter. Now this pedigree is showing the movement of sickle cell anemia. So we can see that just these 2 siblings have the disease, while the other members of the family do not. So that's what you need to know to read a pedigree. If you want to dig deeper, see my videos about how to trace autosomal or sex-linked traits using a pedigree.
Okay, so a pedigree is just a chart that we can look at to see the genetic history of a family. Sometimes, if you ever have to see a genetic counselor, they'll actually create a pedigree with the history that you know, and we'll look at it to help trace the traits that could be passed down. Pedigrees show how a trait is inherited over several generations. and can be used to figure out who might carry a trait versus who has a trait. So, a carrier, you'll notice it asks us down here what a carrier is. A carrier is just an individual who is heterozygous for the trait. So, capital A, lowercase a for the examples over here. So, all of these people, whoops, I did not mean to do that. So, just about all of these people are carriers because they don't have the disease, but because they are heterozygous, they can pass down both recessive traits, and and one of their children can have the disease, whatever the disease happens to be. And a carrier is always going to be somebody who is heterozygous. So as far as the symbols, males are always represented by a square. Females are always represented by a circle. People who are married to one another always have a line between them. Individuals who are affected with the disorder will always have a shaded in square or circle. Unaffected will always be an empty square or circle. So think about, what do you think a carrier symbol would look like? I'll give you a couple of answer choices and you guys tell me what you think. A carrier here is going to be half shaded in, whether it's a square or a circle. In a lot of pedigrees, you'll see them half shaded in. Now, you'll notice if we go back to the previous pedigree that they're not half shaded in there, but if they are half shaded in, that is representing either a carrier male or a carrier female. So I want you to think about, why would a pedigree be useful for a family with a history of disease? And why could we maybe use a Punnett square instead of or use a pedigree instead of a Punnett square? So if we look here, we can see that with this Punnett square, we can see that there is a 25% chance of having the recessive disorder. With a pedigree, we have a little bit better chance of identifying who has the disorder and who passed it down. So if we're looking here, this is Barbie's genes on this side and Ken's genes across the top, and they have a 25% chance of having an affected child, in this case, Sally. But where did Ken get his gene from that was the affected gene? Where did Barbie get hers from? So if we look, Ann and Bill could have had a child who had the disorder. They did not, but unfortunately, they did pass down the recessive trait to be passed down to their grandchild, Sally. So a Punnett square gives us information about an individual child. A pedigree can give us a lot more information about a whole family, and can give us more information about, okay, well, this person doesn't have the disorder, but their parents have the trait and carry the trait. That means that they might be a carrier also. We need to do more testing, kind of things. And then another thing they mentioned in the Amoeba Sisters video, karyotypes. So a karyotype is just a chart that shows chromosome pairs next to one another. And it shows the appearance. You can kind of see that chromosomes are about the same size in each of these. And so because they're about the same size, the karyotype's able to let them see if there's like a deletion on a chromosome, or if there is an addition, maybe something has been added to a chromosome that could be a genetic mutation. You can use a karyotype to determine biological gender. So think about, if this karyotype is male or female. I'm going to drop that as a question in just a few minutes when we are doing the questions for this video. But is this karyotype male or female based on its biological gender? You can also tell if there are too many or too few of any chromosome. So if non-disjunction has occurred in meiosis and the chromosomes didn't separate correctly, then you can see that in a karyotype. Okay, guys, so we're going to go ahead and continue on. So what we need to look at now, as you can see, this karyotype is a male. What we need to look at now are dihybrid crosses. So these are a bit more involved than our monohybrid crosses. So we're going to watch a quick video and then I'm going to show you how to analyze these, because that's the important thing that you need to be able to do, is to analyze it.
traits. Both the males and females can produce 4 different gamete combinations: round yellow peas, wrinkled yellow peas, round green peas, and wrinkled green peas. A Punnett square is created to determine the F2 generation that results from this cross. Of the offspring produced within the F2 generation, 9 are round yellow peas, 3 are round green peas, 3 are wrinkled yellow peas, and 1 is a wrinkled green pea. In other words, a 9:3 to 3:1 ratio exists within the F2 generation. Mendel was able to use this observation to determine that factors assort independently of other pairs of factors. This has become known as the law of independent assortment.
Okay, so like I said, we are going to need to be able to analyze those dihybrid squares. So if we look at this with our Punnett square, they're huge. I am not going to ask you to actually draw these out, but you need to be able to look at them and say, okay, it's asking us how many of the pea plant offspring will have round yellow seeds. So you need to know that round is capital R, yellow is capital Y. So you're going to find every square that has a capital R and a capital Y. So this one has 2, this one has 1 of each, this one has 1 of each, this one has 1 of each. So you can see as you look at this, they all have at least 1 capital R and at least 1 capital Y. So now we're going to go on down to our next row. Same thing. This one has capital R, capital Y. This one does not. So we're going to mark that one out because that one doesn't fit our criteria. It's got to have capital R and a capital Y. This one does. Again, this one does not. Same thing. No capital Y there, which means that it doesn't fit our criteria. Down here, this one fits our criteria. This one does. This one has no capital R, so it does not fit. Same thing here, does not fit. No capital R, capital R, capital Y. And here, no capital Y, no capital R. Neither of them are a capital. So then we're going to count those up. So we're going to count our underlined ones. We've got 1, 2, 3, 4, 5, 6, 7, 8, and then number 9 down here. So there are 9 that fit out of the 16 that set of instructions. And you can see here, they're all highlighted in yellow as well. And that is everything that I have for you for this presentation. Let me know if you have any questions. We'll talk about sex-linked traits, we'll talk about pedigrees, we'll talk about karyotypes, and we'll talk about dihybrid crosses in class for our next class session. I hope you guys have an awesome day, and I'll see you later.